Welcome back to week 2, echolocation in bats, and video 3. So we're moving into bat specializations in sound and neural processing. So after the hair cells have done their job and translated a signal and transduced it into an ordered tonotopic map, now how does the bat actually get information about the environment from that? So where in space and what is the object? And by and large, this is computed by the brain. Okay, so it's able to use all those cues we talked about in video 1: distance, size, azimuth, elevation, and velocity. And we're going to focus on the neural mechanisms for 2 of those today. So we're going to talk about how the brain computes the pulse to echo delay, which is what the bats use to understand distance. Right, how long from when the bat emits a pulse to when it receives the echo tells you how far away an object is. And then the second is how they use Doppler shift in order to calculate the velocity or the speed of the object, the bat, the moth. Okay, and first of all, in the brain, we see this a lot. Size reflects importance. So bats have a very large cochlea relative to their head size. So that's over here. Ooh, that didn't work. Hang on. Let's see. I don't know if I can I point? Sorry, I'm still trying to figure out how Keynote. What's this? No. Can I point with this? No, because it goes away. Okay, well, anyway, I'm gonna have to verbally point, or maybe I'll use color sometimes. That'll work. Okay, so over here, you see that the bat cochlea is a pretty large proportion of its skull. Really, it occupies a large one. You can see humans are over here in gray, and we still have a large cochlea, but we have a much smaller one relative to our brain size or skull size. And this reflects the importance of auditory cues to those 2 different species, taxa. And you can see that in a mouse, these are the sizes of the representations of the sizes of the cortex, the cortical areas devoted to processing a sense. So in a bat, there's a huge area devoted to auditory processing. Makes sense. They're using auditory cues to navigate. A mouse is dedicating a huge amount of space to the sense of smell, and a possum is apparently dedicating a huge portion of their sensory space to vision. And actually, incidentally, you see that in a lot of the different structures on the way up in this pathway. So this is, again, this neural pathway that describes all of the structures that are involved in processing auditory cues in bat echolocation. And so you start down at the bottom, the cochlea and the hair cells, and then information moves up and down as brain areas communicate with each other from there. And so the cochlea synapses those primary afferent sensory neurons onto the cochlear nucleus. And then information goes up from there. And I don't expect you to know all of these things. And in fact, I put the MSO on there. You don't need to actually remember that one. That's important for detecting azimuth, which I decided not to focus on in this unit. So instead, you do need to know the cochlea, the cochlear nucleus, and then it's going to send information up through a bunch of structures that are important and modify the information. And this is going to go through the inferior colliculus, the IC, which is important for a number of different calculations, and we're going to look at some that this is important for. And then through the medial geniculate body, the MGB, before it finally terminates in the auditory cortex, which is, again, is kind of the highest level of sound processing. And from there, the information goes back down to modify perception events. The information can go to other brain regions in order to coordinate complex associative learning or the impact of you know, perception on motor output, obviously super important. You have to have coordination between those brain regions in order to have a successful capture event. But what we're going to be doing is looking at how these brain regions help bats to determine the distance that a prey item is and the velocity or the speed of that prey item. So complex comparisons of pulses and echoes, which is what in video 1 we talked about, are essentially required to determine distance and determine speed or velocity, first require that you have a really accurate distinction of the frequency that is present. So you need to be able to have really strong, good acuity of frequency detection. And looks like some of my slides are going off the bottom there, which is interesting. But anyway, so a brain region where we see really high acuity for frequency detection that then gives information to the auditory cortex, that very important place of perception, is the medial geniculate body. So if we go back, it's that kind of second to the highest structure, the MGB there. The medial geniculate body has two regions. And in this drawing, it's the yellow region is kind of the broader region, and that represents all of the acoustic space, auditory space, the range of frequencies that a bat can hear at all. So of course it hears a lot in echolocation frequencies, but it also can hear in frequencies that you and I can hear. So it can hear, you know, a tree falling over and, you know, a predator walking towards it or something as well. So that's the yellow area. So it's responsible for kind of this broad range of hearing and detecting frequencies. And so if you put an electrode into a cell that was located in, let's say, right here, I'm going to put a green dot. Okay, so if you put an electrode into a cell in the MGB that was located right there, you can see there essentially isolines there of frequencies. So if you listen there and then you played a 40 kilohertz sound to the bat, that neuron would go crazy because it was perceiving a 40 kilohertz sound. If you put an electrode in that, at that green dot in that neuron, and you played a 100 kilohertz sound, it would stay completely silent. It wouldn't fire any action potentials. So this is cool. You can create these frequency maps by putting electrodes in different places, and you can actually map out the tonotopic map in these structures. So that's pretty cool. And we know that that exists because of those labeled lines and the spatial preservation of neural connections from the cochlea all the way through all those intermediary structures to here, the MGB. And the MGB is going to do the same thing and send ordered neurons up to the auditory cortex so that that information is, again, spatially conserved. And so the auditory cortex can understand what that information means. The other thing that's represented here is the pink area, which is called the DSCF, the Doppler shifted CF. In other words, so remember that the constant frequency sound is important for the call, is important for detecting the Doppler shift, and that if the bat flies through space while it's hunting, it can change its call frequency, right? That was the swing experiment. So it can adjust its call frequency to account for the Doppler shift and make sure that the echo is going to come back at exactly the frequency it's supposed to come back at. That's this pink area. So that's essentially the acoustic fovea. That's that frequency that the bat uses most to echolocate with. And so you get this big section that's devoted to a really small range of frequencies. If you look at the yellow, it goes from 10 to 100. If you look at the pink, it goes from 60.6 to 62.3. And what that tells you is that this bat is using right around 61 kilohertz to echolocate and detect its prey. And so it's devoted a large number of neurons to and having really high acuity where it can detect a 0.01 difference in frequency. Whereas in the yellow neurons can probably only detect maybe 1 kilohertz difference. So you get super sensitivity, super acuity in that region. Okay. Computer yells at me. Okay, so you get super accurate frequency discrimination in the medial geniculate body. That was the yellow and the pink area in the previous slide. And what the point of this slide is just to point out that all of the selective filtering that happens by the structures in between the cochlea and the MGB are what gives the MGB its super accurate frequency discrimination. Yeah, that's all you really need to know about what those other structures do, but they're doing a lot. And they're essentially quality control and combining information from different neurons and different hair cells in order to provide a really accurate picture to the MGB. Okay, so now that we know what a frequency is and we know that the brain can detect very minute differences, especially in of the CF, the actual echolocation frequency that's used by that species. Now we can compute something. So we're going to start with distance and how they can compute this pulse to echo delay. So when they give the call, it bounces off something, comes back, the time that that takes. And there's a region in the auditory cortex, which is the green region here, called the FM area. And if you put an electrode into a cell in the FM area in this region of the cortex, they get really excited when there's a particular delay between the base frequency, FM1, and the returning echo pulses of some of the other harmonics in the call. So we know that it's the region that says, okay, here, this is a certain amount of delay, and that delay can be perceived as a distance away. And we'll talk about that. So here's a call. So this was from a prior slide. And what you see is that the CF1 down here at the bottom, and then you can see the frequency modulated part of the call is at the end. And so for the returning echoes, they're going to use that FM part, and it's going to compare the first call given, so the pulse, with the time it takes for the echo to return. And so you can see the delay is represented by the black arrow on the bottom of the figure there. That's about, 8 milliseconds. So that object is, it took 8 milliseconds for the echolocation to travel out, hit the object, and to come back. And that delay should be essentially identical for all of the harmonics that are in that pulse. And so what happens is the lowest harmonic, CF1 there, the brain pays attention to the timing of that call. When they give CF1, it's listening. So when a bat emits a call and makes that chip that we can't hear, obviously, because it's a different frequency range than us. Essentially, the neurons, the hair cells, are detecting the start of the call that the bat made, and then they're listening to the 63 kilohertz echo for FM2, the 90, I'm going to point. Okay, so it's going to compare that timing to when these are detected. Oh, and I actually, let me redo that, because I just drew that as the CF. These are FM comparisons. So basically it's going to compare the call given, this FM sweep, to the return of the echoes. Okay, so that's for distance specifically. There's a different, the Doppler effect is going to use those CF that I was just pointing to. But for distance, the FM sweep is the more accurate, or I guess just the better thing to use for range detection. Okay, so cells in this region are excited by a particular pulse echo delay, that delay between when the call is given and the echo returns. So a neuron's position in the FM area of the cortex thus basically means or signifies a distance. So again, you have place equals something meaningful. And in this case, in this area of the cortex, place refers to distance. So for this, which neuron, the blue, one represented by blue or yellow, these are just neurons that are in the cortex in this region, would represent an object that is closer to the bat? I'll give you a second to think about it. So hopefully you came up with blue. So the blue object is closer to the bat because there's a shorter echo delay. So if you're listening to a neuron at that blue star, and the bat makes a call and the echo returns in 2 milliseconds, that blue one's going to light up and the bat's going to know that object is close. If you're listening at the yellow star into a neuron with an electrode, and you play, the bat makes a call and the echo delay comes back as 10 milliseconds, then the bat knows that the object that it hit is much further away. And specifically, it's going to actually equate to a precise distance, depending on the species and the call. So this tells you that the processing pathway all the way up and terminating at this region in the cortex can calculate that delay. But where is it actually calculated? How does that blue neuron, why does it fire when the echo delay is 2 milliseconds? You know, what happened in the brain below that? So a reminder here that not sure why that slide was blank. I hope hopefully nothing isn't showing up. So we have our pathway here from the cochlea all the way up to the auditory cortex. And it turns out that, oh, you know what? Hang on. I bet you this is a, Okay, we're gonna see. There might have been something critical missing there, but we're gonna find out. We'll come back to it if there is. Okay, so here's our pathway. We have our organization from the cochlea up to the auditory cortex. And we know that for distance detection, once you're in the auditory cortex, in the FM area, that cells light up really specifically depending on the delay. But what is calculating the delay? So it turns out that if you probe each of these brain regions with an electrode, that the neurons in the medial geniculate body, in the MGB, again, are the first ones in the whole pathway to respond to specific pulse echo delays. So basically, by the time you get to the MGB, there has been some determination of what the delay is. So what that means is that something further down has to be kind of leading to the MGB to have that specificity, that delay specificity. And what we know is that neurons in the inferior colliculus show an interesting pattern. So they fire immediately in response to echoes. So the echoes, when an echo hits the cochlea, the inferior colliculus fires right away, the neurons in the inferior colliculus. But they delay their response to the calls. I wrote that wrong there. So I'm going to correct it on the slides here, and hopefully I remember to correct it in this is the pulses. Okay, so the call itself. So the echoes don't have a delay. The pulses do. So when the bat makes a call, a click, you actually see a delay to when it shows up in the inferior colliculus, at least in some cells. And what these neurons are doing in the inferior colliculus is they are constituting what's called delay lines. And this is going to be important for determining the pulse echo delay in higher levels in the MGB. So hang on, we're getting there. So here it is. So what we see is that divergence of action potentials from a single cell in the inferior colliculus onto neurons in the MGB are, and then adding in delay lines, allow for coincidence detection in MGB neurons. So this will all make sense, hopefully. We might have to go over this in office hours again. But what you have is, you can imagine there are different neurons in the inferior colliculus that are listening to different frequencies. And we know that the harmonics are all different frequencies, right? They're stacked on top of each other and they're all different frequencies. And so when the pulse is made, the FM1, okay, so the lowest frequency in each of those harmonics, it is detected by the inferior colliculus here. And those neurons are firing action potentials. So basically all of these dots represent synapses. And so neurons are transmitting the information down the pathway. And we're going to pay attention to kind of the left half here. The right half is, you can consider it similar, but it has an even longer delay. So here comes the signal. It hasn't been delayed yet. And when it gets to these points of divergence, the fastest, one of the pathways doesn't have any more synapses, right? So it's fast. And it's essentially offering no delay. Okay, so that signal basically reaches this MGB neuron the fastest. The other pathways, so like this one, synapses also onto an extra step, like an extra neuron that slows that signal down just a little bit before it gets to those. And sorry, this first one is diverging onto 4 MGB neurons. This next one onto the next 4. And basically, this is no delay. This one has 1 extra delay. This pathway went through 2 extra delays. And this pathway went through 3 extra delays. So we're talking about time delays here. So basically you have one single pulse, right here. This pulse at the very start of that whole pathway is diverging. So it's originating from one event. And it's detected that action potential from that one pulse from this cell in the inferior colliculus is passed through all these delays so that you have the signal arrives at different times. It arrives, so we're going to say that this FM1 pulse happens at time 0. So you have these cells are getting a signal at time 0. These cells at time 0 plus 1. These cells at time 0 plus 2, and on down the right. Time 0 plus 3. Okay, so now you have essentially kind of a zipper effect where if this MGB neuron is going to receive a neurotransmitter signal from the inferior colliculus first at the blue, and then it's going to zipper down towards the right as those delay lines slow it down a little bit. The echo coming in from another, remember this is a different frequency, so it's coming in from a different inferior collicular neuron, never has a delay. So it's coming in and it's essentially always synapsing onto these neurons at the same time. So, but the echo is delayed in real space out in the world, depending on how far away the animal is. So we're going to call the echo time is the distance, right? It's the timing at which the echo comes in reflects how far away the object is. But you don't know what that distance, what that time is, unless you compare it to the original call, because that's the start of the timer. So you have the end of the time event and the start of the time event. And these delay lines are what allows you to know how long was that delay. So if the echo comes in really fast, okay, the object's super close by, then all of the echo all of the echo neurons are going to release neurotransmitter at the exact same time. But only one subset of the delay lines from the pulse are going to release neurotransmitter at that exact same moment. So if it's close in space, and there's essentially no delay, if the echo is close, then when the bat made the call, it might match up in time to when the signal from the call is quickly occurring, right? So because there's no delay, so it's almost immediate. And so this here, when these neurons fire, these MGB neurons, or detect neurotransmitter from both the echo and the pulse at the exact same time, then those MGB neurons will fire an action potential. All the other ones are only getting neurotransmitter from the echo neurons. They're not getting neurotransmitter yet from the pulse because of the delay line. And so if the target is close by, then the only MGB neurons that will fire will be these blue ones. If the object is further away, let's see, what color have I not used? I've used most of my colors. I'll use, we'll just say this. So if the object is further away, remember the echo still comes in at the time it comes in, but it's coming in a little later in time. And so the coincidence might be that the blue neurons from the pulse call have already released transmitter. Then the green neurons release transmitter. It's still not coincident with the, let's see, I'm actually going to back up because I think I can. Let's do this. We're going to take away that fast response. Because what I want to show you now is if the object's further away. Okay, so if the object's further away, oh no. It's going to keep going. Okay, there it stopped. Did it? Yeah. Sorry. I guess it wasn't as fast as I was. Anyway, you remember what this all looks like. Okay, so that's all happening at once. But, okay, so those are the lines. So if the object is further away, then the time for the echo to actually be perceived by the ear is going to be a little slower. So you might have already seen some neurotransmitter release from the pulse, which was the original call, from those guys. And then there's a little bit of a delay. And then this group fires. Still, the echo hasn't come back. And then all of a sudden, maybe this group fires. with that delay at the same time that these guys fire. Okay, so that echo finally came back after the second delay. So now you know that the object is that distance away because these MGB neurons, these purple ones, are going to fire an action potential because they had coincidence detection, whereas the other ones hadn't. Okay, it's complicated, I know. Hopefully you're understanding that. You can read the book to help you to understand that as well, and I can go over it again in office hours if you're having a hard time. But basically it comes down to the MGB neurons are the ones that are detecting the coincidental firing of or signals from the echo and the pulse, which may or may not be delayed. And that coincidence then can give you information about how long it took for the echo to come in. What that means is that you have to have coincidence detection. And so this comes back to some of that background where they're only going to fire an action potential if both the red and the black are firing at the same time. And I said red and black, but of course I was using different colors, but you can see the black line coming in from the echo on this figure and the red line coming in from the original call pulse. And so when those two things coincidentally fire onto an MGB neuron, and then you get temporal summation and the MGB neuron fires. And now you have calculated, again, the space in the MGB neuron is going to determine where it is in that structure is going to tell the cortex what the delay is. So the blue down there at the start, okay, that was essentially no delay in the pulse. And so that is a very short delay for the echo. And therefore that would translate to this object is very close to me. The further you move down the delay lines, the longer it took for that echo to come back. And so you see the yellow one, that represents some distance away, and the green is the furthest away. So again, you have a spatial organization, in this case, for distance. Super cool. Okay, so now we're gonna touch on Doppler shift and speed. So this is a different region of the brain. So here is this representation again, and we had just been talking about the green FM area that compares the delay lines. And now we're gonna move to the CF area, the orange region in here in the cortex. And this region responds really precisely to differences between the CF harmonics echoes and CF1. And this is going to give us information about the Doppler shift. Ooh, this slide is a bear. So, sorry. So the frequency, so remember when a bat gives a call, the harmonics are all stacked up on each other. So the call, for a CF call, it's going to be, so we have that graph that's like this with frequency here and time here. And the bat's giving a call, and that's CF1. But then there's harmonics, CF2, CF3, CF4. Okay, so the harmonics, and then they're going to echo back a little bit later in time, right? So maybe like that. So that's the echo coming back. The frequency of that harmonic, it should be the exact same as what was emitted. But Doppler shift will cause it to change slightly. Delete this so we can read again. Okay, so Doppler shift, if the object is moving towards the bat, so remember this is the car now moving towards or away from the person. If the object is moving towards you, then that causes, towards the bat, that causes the sound to appear slightly higher in frequency because it's pushing those sound waves closer together. And then when it's going away, the object's moving away, and so it's pulling the sound waves further apart than it should be. And so the frequency should be the same as what it emits, just bounced off and comes back. But if the object is moving, then it's going to bounce back at a different rate, and that's going to change the frequency of the sound. The faster the object is moving, the more the larger the Doppler shift. Okay, so here's an example. So let's say that a bat produces a 30 kilohertz CF1 and then a harmonic above that that's at 60. Okay, so just like I drew those red lines stacked on top of each other, the first one's 30, the second one's 60. If the harmonic returns at 60, the bat knows that the object is not moving. But if it comes back at 61 kilohertz, then the Doppler shift is 1 kilohertz, higher, which translates to an object moving towards the bat at 2.8 meters per second. That's all physics. And what the bat needs to be able to do is to compare the outgoing frequency of the call to the incoming frequency of the harmonics. And that's what it needs to be able to compare. So neurons in the CF area of the cortex respond to really precise frequency and amplitude differences. So note that there's also a loudness component here, amplitude. And we're not really going to talk about that. We're going to talk more about the frequency component. But know that amplitude matters too, how loud, how many decibels the sound is. So here's a graph that shows loudness on the y-axis and frequency on the x-axis. And so in this example, this is a neuron. So again, you're in that orange area of the cortex in the CF area. If you put an electrode into a neuron there, it's going to have a really precise frequency that it will fire action potentials to. And specifically, it's going to have, so this neuron, for example, in this graph, is going to fire an action potential when the frequency is of the CF1 is about 20. So actually, if you look at the whole wide range there, those are the frequencies of CF1. So it'll get excited if there's CF1 between 22 and 26 kilohertz at the same time that there's a harmonic of 60 kilohertz. So each neuron that you listen to in that CF area will have a different preference for that. There might be a different neuron that responds to maybe these exact same CF1s, because for a species that should be fairly similar, but maybe it responds when the harmonic is Doppler shifted up. And maybe a third neuron responds only when the Doppler shift is down. Okay, so there'll be a neuron for literally every single combination. And what that's suggesting is that, again, in the processing, kind of before you get up to the cortex, something's allowing for this kind of level of, or pairing, I guess, of the two CF1 and the echo frequencies. So the position of a neuron in the CF area represents the velocity or the speed of the object then. So if you have a neuron that's at the blue star, that represents a larger Doppler shift. So there's a bigger difference between what the expected harmonic is and what it was. And though when it fires, it means the object is moving fast and in a particular direction. And if you get a neuron in a different region, then it'll have a different speed and direction associated with it. And so in this way, the cortex can perceive velocity and tell a bat how fast and in which direction it's moving. Okay, so very briefly, I want to just address how do the moths escape? Because we focused really heavily on the bat side of things. Basically, they have ears too. So inside their air sacs, there's a tympanum, just like mammals have. So it's a membrane that vibrates. And there are two receptors. This is a pretty simple system, as many invertebrates have, and it's the beauty of studying invertebrate neurobiology. There's an A1 cell and an A2 cell, and these are two different neurons, sensory receptors. Oh, what just happened? And they respond differently. A1 responds to lower decibels, so quieter, and less frequent pulses. So this represents a distant hunting bat. And when that A1 cell fires, the moth moves away from the ear that detected it. So a moth has multiple ears and they point from the abdomen in different directions. So if one ear detects the sound but the other ear doesn't, then it's going to move away from the direction that that ear detected the bat. So in other words, it gives it a way to move away from the bat before the bat's close. The A2 cell only responds when there's very loud decibels and when very frequent pulses. So in other words, a close bat that's using a terminal phase echolocation. And when this happens, it's a reflex that immediately causes the moth to go into a kind of a spiraling dive and try to evade the capture by the bat. Oops, I want to pause that. So the last thing we'll end on is just a really cool anti-predator moth adaptation that moths have evolved in order to jam the echolocating sonar of the bat. Okay, maybe I have to go.
Slow motion night vision technology allows us a glimpse inside the bat's world. Sonar detects insects in complete darkness. A big brown bat targets its prey and locks on.
Like sound effects. Catch by the wing.
But new science reveals that the tiger moth has a secret weapon. Ultrasonic sonar jamming technology. Its high-speed clicks are slowed down so we can hear. When a bat locks on, this moth fights back.
Okay, so just a cool example of some of the things that moths have evolved to try to evade bats. And then bats respond to that kind of thing by producing echolocation clicks in frequency ranges that the moths can't detect. So the moth doesn't even know that it's coming. And that's stealth echolocation, kind of anti-predator prey, evolutionary arms race in a sense. Okay, so my guess is you're going to have some questions down the road about some of this stuff, and that's fine. We can talk about it more in office hours. That's the whole point. So we'll see you guys soon.